Deep beneath the cold ocean surface, a giant Pacific octopus propels itself across dark granite rocks. Inside its body, liquid flows not red, but clear blue. This distinct coloration stems from copper atoms binding oxygen, an evolutionary adaptation made hundreds of millions of years ago. To drive this heavy, copper-laden fluid efficiently through its body, a single central heart is insufficient. An octopus relies on 3 distinct hearts working in harmony. 2 branchial hearts pump blood exclusively through the gills, while a larger systemic heart circulates oxygenated blood to organs. This intricate system enforces a dramatic physical penalty every time the animal moves. When an octopus swims using jet propulsion, the main systemic heart stops beating entirely. Pushing high-pressure water through its mantle funnel physically compresses and arrests the central cardiac muscle. Because its primary systemic heart stops during active swimming, the animal exhausts quickly. Octopuses rarely swim across open water for extended distances. Instead, they crawl along the seabed using their flexible suckers, reserving jet propulsion almost exclusively for sudden, brief escapes from dangerous predators. Human blood relies on iron housed within red hemoglobin to trap oxygen, imparting a vivid crimson hue. Octopuses abandoned iron early in their evolutionary lineage, opting instead for large, floating protein complexes known as hemocyanin, which rely entirely on paired copper ions to bind oxygen. When hemocyanin binds to oxygen molecules, the copper ions oxidize, shifting from transparent fluid to brilliant blue. Unlike hemoglobin contained neatly inside blood cells, hemocyanin floats freely in the plasma, making octopus blood thick, highly viscous, and remarkably energy-intensive to pump. Moving dense blue liquid through narrow vessels demands immense muscular force. Without 3 dedicated hearts generating targeted pressure gradients, oxygen would never reach the dense muscle tissues or the massive, energy-hungry central nervous system distributed throughout the mantle and arms. For centuries, early European naturalists misunderstood the internal anatomy of marine invertebrates. Dissections of dead cephalopods often revealed pale, clear fluids in their vessels, leading researchers to conclude incorrectly that these soft-bodied creatures lacked a true, functional circulatory system altogether. In 1878, Belgian physiologist Léon Fredericq performed precise chemical analyses on the blood of Pareledone muscata, a Mediterranean octopus. He noticed that pale fluid harvested from living animals turned brilliant blue upon exposure to air, proving an active oxygen-binding pigment was present. Fredericq named this newly isolated respiratory protein hemocyanin, derived from the Greek words for blood and blue. His experiments demonstrated conclusively that copper, not iron, served as the functional core of the molecule, establishing a fundamental new concept in comparative physiology. Subsequent comparative anatomical studies revealed that cephalopods had evolved a fully closed circulatory system, unlike their distant snail and clam relatives. Snails retain open sinuses where fluid bathes organs directly, powered by a simple single-chambered heart operating under very low pressure. The ancestral condition is visible today in the chambered nautilus, a primitive cephalopod retaining an external shell. The nautilus possesses a simpler circulatory layout with far lower metabolic output, serving as a living bridge between slow-moving gastropods and agile modern octopuses. As ancient octopuses shed their heavy protective shells around 275 million years ago, speed and agility became vital for survival. The loss of passive armor required rapid athletic bursts, driving the evolution of auxiliary branchial hearts to drastically accelerate oxygen uptake at the gills. This evolutionary transition created one of the most specialized vascular networks in the animal kingdom. 3 independent cardiac pumps working alongside copper-based blood enabled soft-bodied octopuses to become dominant predators across virtually every depth zone of the global ocean. In the freezing waters of the Southern Ocean surrounding Antarctica, the polar octopus Pareledone charcoti survives in water temperatures dropping to -1.8 degrees Celsius. At these extreme thermal limits, standard physiological processes fail, yet this species thrives on the icy sea floor. Freezing cold water dramatically alters chemical kinetics within biological systems. As ocean temperatures drop toward freezing, standard hemocyanin holds onto oxygen with extreme tenacity, refusing to release it to surrounding tissues. Without physiological adaptations, a polar octopus would suffocate despite abundant dissolved oxygen. To overcome this thermal lock, polar octopuses alter their genetic gene expression. Research reveals that Pareledone charcoti produces custom hemocyanin proteins modified at specific amino acid positions, lowering oxygen affinity at low temperatures so oxygen unbinds smoothly into shivering muscle tissue. Furthermore, polar species maintain exceptionally elevated concentrations of hemocyanin in their bloodstream. Up to 40% more blue protein circulates through their vessels compared to warm water tropical octopuses, effectively compensating for sluggish chemical reaction rates in sub-zero polar environments. This high protein concentration turns polar octopus blood remarkably thick and sludgy. Pumping this viscous fluid through cold, stiff blood vessels places immense mechanical stress on cardiac muscles, requiring precise pressure boosts generated by the twin branchial hearts situated at the gills. The branchial hearts contract forcefully, driving dense blue blood through delicate, highly folded gill filaments. Here, carbon dioxide diffuses outward while fresh oxygen binds to copper centers before the blood travels inward toward the systemic heart for central distribution to the body. This delicate thermal balance allows octopuses to inhabit diverse ecological niches from tropical coral reefs to abyssal polar trenches. Yet this heavy reliance on custom copper proteins leaves them uniquely vulnerable to rapid environmental shifts in global ocean temperature and chemistry. Despite its adaptability across temperatures, hemocyanin is fundamentally less efficient than hemoglobin under normal atmospheric conditions. A single human hemoglobin molecule carries 4 oxygen molecules, whereas hemocyanin binds oxygen far less compactly, requiring much higher blood volume to transport equivalent oxygen loads. This efficiency gap means octopuses operate remarkably close to their maximum physiological capacity even at rest. They possess minimal aerobic scope, meaning any sudden increase in physical exertion or environmental stress rapidly pushes their 3 hearts to their maximum biological limits. The functional efficiency of hemocyanin is also hypersensitive to changes in surrounding fluid acidity. As ocean waters absorb excess atmospheric carbon dioxide, seawater pH levels drop, a process known as ocean acidification. Lower pH destabilizes hemocyanin's chemical structure, severely reducing its oxygen-carrying ability. When warming ocean temperatures coincide with dropping pH levels, a severe physiological crisis emerges. Warm water holds less dissolved oxygen while simultaneously accelerating the octopus's metabolic rate, forcing all 3 hearts to pump much faster just to maintain baseline cellular function. Laboratory experiments conducted at marine research stations demonstrate that elevated water temperatures cause severe cardiac arrhythmia in octopuses. As thermal thresholds are exceeded, the systemic heart begins skipping beats, collapsing the pressure gradient required to nourish vital internal organs. Unlike fast-swimming fish that can migrate vast distances to seek cooler deep-water columns, benthic octopuses are often bound to localized home territories. Their physiological dependence on high-pressure copper blood severely restricts their capacity to adapt to rapid, human-induced environmental shifts. This narrow physiological tolerance reveals the ancient evolutionary trade-off made millions of years ago. Pairing copper blood with 3 separate hearts enabled remarkable intelligence and physical agility, but established strict biological boundaries that modern changing oceans are now actively testing. Evolutionary biologists often question why octopuses never transitioned to iron-based hemoglobin during their long evolution. The answer lies in historical contingency. Early ancestral mollusks developed hemocyanin in shallow, copper-rich primeval oceans where iron was far less bioavailable in soluble, usable forms. Once a complex closed circulatory system evolved around hemocyanin proteins, shifting fundamental blood chemistry to iron would have required rewriting thousands of developmental genes simultaneously. Evolution modified existing structures instead of starting fresh, optimizing the triple heart layout to fit hemocyanin's limitations. This massive metabolic investment yielded astounding cognitive results. Oxygen continuously supplied by 3 hearts powered a decentralized brain containing 500 million neurons, enabling complex problem-solving, dynamic camouflage, and physical tool use unmatched by any other invertebrate group on the planet. Yet the heavy energetic tax of powering 3 hearts and dense copper blood comes at a fatal cost to longevity. Most octopus species live fast and die young, with lifespans ranging from just 6 months in smaller species to 5 years in giant Pacific octopuses. Reproduction marks the absolute end of an octopus's life. After laying her eggs, the female stops feeding entirely and undergoes programmed cellular breakdown, while males deteriorate rapidly after mating. High metabolic burn leaves 0 energy reserves for somatic repair or long-term cell maintenance. In their brief lives, octopuses demonstrate how natural selection can build extraordinary biological complexity through unexpected chemical pathways. 3 hearts continue beating in dark ocean depths, driving clear blue blood through a body designed for brief, intense, and brilliant survival. As global ocean chemistry shifts faster than at any point in human history, marine scientists watch these creatures closely. Whether this ancient, copper-powered circulatory machinery can adapt once more to a warming planet remains an unresolved question resting silently on the cold sea floor.